Okay, so I'll be very quick. So I'm Michelle van der Bank from South Africa. I'm head of the African Center for DNA Barcoding, but I also have a position at the university lecturing quite a bit. Um, my interest is mostly plants, but recently I've moved a bit more to the zoological side, but actually I have my qualifications in zoology, so I'm just going back to my roots. So our focus is mostly Africa. We don't go out of Africa, actually mostly Southern Africa, and we've put up some nice collaborations now in Mozambique where we're going to collect some tree species. I've been involved, or the lab, in barcoding almost since the start, so a long relationship with iBOL. The numbers. 98,000 sequences, but it only represents about 11,000 named species. I'm happy to say half of it is plants. We have about 22,000 species of plants in South Africa, so almost a quarter of the work is done. The rest, I'm not so sure how many animals and fungi. I think there was a prediction about 100,000, so a lot of work still that we need to do. So South Africa has signed MOU with the first phase of iBOL. I think it was in 2011 or 2012. At that point, we had a steering committee that steered the operations of SA Bolt in South Africa. We're in the process of signing the MOU. I don't know whether you got the MOU yet.
Yeah, it's edging closer.
So I have been in contact with Sandy last week, and the problem is with government. And it's Africa, government takes a long time. So DST will sign it off. We have meetings with them, and we actually drafted a strategy for barcoding in South Africa, a new strategy, so it will be signed, so don't worry. So basically, Sandy will lead SA Bolt in South Africa. We won't have a steering committee for barcoding anymore. We will fall under the Foundation Biodiversity Information Program, where Jenny went there earlier this year. We have quite a number of researchers claiming that they're involved in barcoding, about 80 in the country, which represent about 25 institutes. If I say researchers, I'm including students in it. So 25 institutes that's involved in it. And our funding mostly comes from the National Research Foundation under this FBIP program, which is quite nice. This program started paying out funding in 2013. They have large projects where you get about 6 million over three years, and then they have several small grants, and it's actually quite easy to get this money. And what's nicer about it, they fund students. So student studies is funded through them. So basically, if my students apply, they all get money, and I think this is for everybody, students that's applying for this. Now, we have lots of future plans, but I think the most important thing is to get additional funding in. We don't have a shortage of money, but it's always better to get more money in. But also to, actually, we were quite strong at some point, the node, and then we took a little dip, and it's now to get the node up and running again. So this would be our focus the next year or so. And like I said, we have already drafted the strategy for where we're going and which institutes will be primarily involved in it and what the task is, what we will do. So for the projects that I want to share with you, I've chosen two projects, one that's funded by government and one that's funded by this FBIP program, just to give you an idea what kind of program are funded at the moment in South Africa. There's many projects, and tomorrow I will talk about the Malays projects and the projects that's running in the Kruger Park at the moment. So the first one is the so-called biogaps project, which is quite an interesting project. It is in the Karoo area in South Africa, and this is the area that they actually identified for fracking. So there must be a biodiversity survey done before Sasol can go in or to get scale gas. So I think that that's not going to happen in any case in the near future. So we have a large group of collaborators on this project. It's led by Sanby. We have 2 barcoding labs that's involved in it, and it's the only 2 really big labs in South Africa, which is the National Zoo, which handles the zoo. Basically, they do the fish and the mammals, and all the rest, like the invertebrates, comes to ACDB. So it's the 2 big labs doing barcoding at the moment. So something that I want to point out, and I think this is something that we as a group in South Africa will have to look at very carefully, is the amount of money is 6 million ZAR, South African rand. So I think it's about 600,000 Canadian dollars over 3 years. And if you look at the target groups here and the amount of barcodes that has been produced for this amount of money, it's only 1,000 barcodes. So it's not a lot of barcodes for that amount of money. But what we try through this program is that, for instance, if samples is sent to UJ or ACDB, we force the researchers to load their data onto BOL. We don't send the data back to them. They have to access it through BOL. So in a way, we can control that people are actually doing barcoding if they get funding for that. So like I said, this is just one of the projects. The latest project that was funded is quite a sexy title, Snapshot Safari. This is the latest project. But mostly it's camera trapping, but they will catch some small mammals to be barcoded. And then the second project that I want to mention is quite a successful project, is the Barcode of Wildlife project. I heard somebody talking about wildlife forensics just now also, Philippines, I think. And this was a project that was funded through iBOL with a Google grant. And what's very nice about this project, when the funding stopped, government, the Department of Environmental Affairs, took over the project. And they're actually putting a lot of money into this project. The only thing is again, like the previous project, is we don't produce so many barcodes. So for the initial phase, we targeted 210 priority species, 800 closely related species. So now we really have to go further and barcode some more species here, but you can see the taxonomic groups here. And it was interesting that plants actually make up the most traded species. Mammals, I think, was the least. So what's also very successful in this project, it's really brought barcoding to the attention to government. And like I say, government is quite sponsoring a lot of our projects at the moment. They're sponsoring for the past probably 8 years the invasive project. We finished the phase with all the invasive plant species. We're moving next year into animals, which might be a bit more tricky, go fishing. So we're really getting good support from government, and I think we can actually push them for a bit more support to continue. So if we look at the SWOT analysis, and I was actually, when I was doing this, I sent out the form to several researchers in South Africa, and I was very surprised to see that people still put down that they're afraid of barcoding, that there's a big resistance to barcoding, even though we did lots of training through the country. Maybe there's a change in researchers, so we will have to do a lot of training again. But the strength is, for us, is that we have 2 very strong barcoding labs. We do have enough funding, even though we should look for more. And opportunities here. Oops, my ways. The NRF, our National Research Foundation, has just awarded a grant of 50 million for the next 2 years to build up a central collection facility, and this will be run by Michelle Heimer, which is very good for us because she's very pro barcoding, and she has already told me that a lot of those taxa will be barcoded, so we could get some more taxa in through there. And I think that's it.
Thank you.